I'm going to focus on a very particular subject, and that's mutations, because mutations are a key problem with evolution, and I'm, in a certain sense, uniquely qualified to talk about them because I spent most of my undergraduate years, my graduate years, and my time doing research outside of the university working in the field of genetics. So I'm actually more qualified than Richard Dawkins to talk about this particular subject, which I like to point out to people. So my CV is boring, but I did work for a short period of time at the Genome Sequencing Center at Washington University in St. Louis, where they did the Human Genome Project. And I worked for a brief period of time studying developmental genetics in a model organism. So all of that is going to kind of come into play during this presentation. And hopefully I will make it understandable to the average person who has not worked in a genome sequencing center. That's my goal. All right, so when you think about evolution, it's important to kind of understand what's being presented, and then you can really evaluate whether or not it is scientifically tenable. So the basic idea behind evolution is that in a given population, you have too many individuals for the environment to support. So you have an overproduction of individuals. Those individuals will be of a variety of different types. They'll have different heights, different weights, different color patterns. There'll be a number of different genetic differences. Some of those differences will be more adapted to the environment than others. And then you get what's called natural selection. So the individuals that are less well adapted to live in the environment will be weeded out of the environment. That's competition. Then over time, the population adapts. Individuals don't adapt. It's a common misconception. The population changes over time as new individuals are born and old individuals die, particularly as old individuals die without reproducing. And then you have a change in the population over time. So you're given, students are often presented images like this to show that evolution is true. And if you look very closely at this image, you might notice something important, which is that the giraffe doesn't become anything other than a giraffe. This tends to be overlooked by evolutionary textbooks. So this image is trying to tell you sort of two competing things. One is that mutation and natural selection are how we got all the variation in living things, but what they're actually showing you is how mutation and natural selection reduced variation among living things. That's what you're actually seeing in the image. So we're going to sort of look at this, this idea of mutations and this where mutations can actually take us. So this matters immensely because mutations are necessary for evolution to be biologically tenable. Okay, so in order for natural selection to work, I have to have variation in the population. In order to have variation in population, I have to have mutations. So if I can show you that mutations do not lead to the kind of changes that we need to go from a microbe to a microbiologist, I have effectively biologically neutralized the theory of evolution. So that's why I chose to focus on this particular topic today, because it's such an important linchpin. There's a lot of other things that I'm going to cover on Saturday in Charlotte, if anybody wants to trot over to St. Thomas Parish. I'm sure Mr. Owen can tell you a little bit more about that at the end. But today I'm just going to focus on mutations. So to understand mutations, an analogy is helpful. So you can think of a genome, this is the whole set of genetic information in an organism, as an instruction manual. And that set of information has to have all of the information necessary to build the organism. So in this case, we're going to build a giraffe. So if you were going to look at the instruction manual for a giraffe, that's approximately 3 billion nucleotides or 3 billion base pairs. You can think of those as letters. You'd have to look at 10 letters per second, 40 hours a week for 68 years to view all of this information, just to give you some concept of the size of what we're talking about here. Okay, so that's all the information that you need to make a giraffe. And evolution claims that there's changes to that information over time. These are mutations. A mutation is a change to the genetic code. And as I accumulate more and more and more changes, eventually, with the magic of natural selection, I will get a better giraffe. This is how evolution is supposed to work. My contention is that how evolution actually works, as I accumulate more and more and more mutations over time, is that I end up with a non-functional giraffe. Okay, so mutations, I'm going to contend, do not build up information, they destroy information. And if that is correct, they're not a good mechanism for Darwinian evolution. So we're going to sort of turn this on its head and look at what would be necessary for mutations to be the mechanism of evolution. And then I'm going to address each of those points individually. So the first point is that most of that instruction manual, most of that 3 billion base pairs should be useless. Well, why? Well, if I'm changing it all the time, and I'm changing important stuff, that's a problem. That will lead to a dead giraffe if I change something that the giraffe needs in order to build its body. Right? So if most of the instruction manual is useless, then mutations aren't really a problem. Changes aren't really a problem. Right? So this second point goes with the first point. Most of the changes should be harmless. They should be neutral mutations. Okay? So if most of the instruction manual is junk, and most of the changes are neutral, and a few of the changes are good, then maybe I can have the raw material for the magic of natural selection to do its thing and improve an organism. So I also have to have the accumulation of these mutations leading to new kinds of organisms. Okay? So most of the instruction manual should be useless, most of the changes should be harmless, some of them should be good, and putting them all together means I get a new kind of organism. So those are the 4 points that we're going to look at in detail today. So the first question I have is how much of the DNA is useless? Now, you may have heard the term junk DNA. The term junk DNA was coined in the it was formalized in a paper in 1972 by Susumu Ono. And he basically made the contention that most of the genetic information, most of that instruction manual for an organism has to be junk, because we're seeing mutations, lots and lots of mutations every generation. And if that many mutations were occurring and they were occurring in areas of the genome that were important, we should see error catastrophe in the genome over time. We should see something called genetic entropy, that over time the information in the genome decays. It doesn't get better. So he said, well, most of DNA has to be junk. And then in the 1990s, that idea was cemented in the popular imagination when we realized that only about 2% of your instruction manual codes for proteins. So only about 2% of your DNA is genes. And I'm sure you've heard the word gene, but it's been a long time since high school biology. So let's talk about, for a second, what a gene is. So you can think of a gene as a particular paragraph in the instruction manual that's super important. It codes for a very specific molecule that you need in your body, and those molecules are proteins. Proteins do basically everything that gets done in your body. If there's a chemical reaction that has to happen, you need a protein. If there's something you need to build, you need a protein. If there's something you need to move from one place to another, you need a protein. Now, we think of proteins sometimes as energy. My students made this mistake a lot. That's the only thing they're not for. They're for everything else, structure and function. So if only 2% of my genome is genes, well, it seems like the rest would probably be junk, right? And this is hard to sort of conceptualize in a very abstract way, so I'm going to give you an analogy. If I handed you a box of laboratory equipment, all the important pieces, could you teach a chemistry class?
No.
Well, why not? Well, I don't know how to use this equipment. I don't know when to use it. I don't know what to put in it. I don't know, because there's a lot of regulatory elements involved, right? I need to have a lesson plan. I need to know when I should be doing stoichiometry. If you teach that before you teach, molecular mass, you're going to have a giant problem. Your students are going to have a clue. They don't usually anyway, but they're certainly not going to have a clue what you're talking about. Okay, so the genes code for the proteins, and the proteins are super important, but they're just the materials that are involved in life processes functioning. There's no information in the genes about when or how or where. And if you think about this in a body, right, you have that whole instruction manual, you have 3.5 billion base pairs, so it'll take you even longer to look at them all. But all 3.5 billion base pairs are in every single cell in your body. But your eyes are very different from your skin, which is very different from your liver, which is very different from your lungs. So certain proteins have to be made in particular places at particular times in development and in growth and as an adult for your body to function in its multicellular complexity as a single organism. So there's a lot of regulatory information. So really, our earlier analogy of an instruction manual is not quite adequate. The genome is more like an operating system than it is like a library. And if you think about your operating system, if Skype and Word and your email and everything else that could possibly run on your computer all opened at the same time, that would be really annoying, and you'd never be able to get anything done. You'd spend your first 30 minutes closing Windows before you could even start a project on your computer. So everything has to happen in the right way, at the right time, in the right amount, and that requires a lot of regulatory information. So theoretically, we would think that most of the DNA actually wouldn't be junk because there's a lot of information required to regulate that 2% of genes. And it turns out, experimentally, that's true. So after the Human Genome Project, there was a project called ENCODE, the Encyclopedia of DNA Elements, and they looked at the genome sequence that was generated during the Human Genome Project, and they assigned at least 80% of it meaningful function. So we've gone from 98% junk to maybe 20% junk, and I'm going to explain in a second why I don't even think there's 20% junk. But why did they do this? Two basic reasons. Most DNA is transcribed, and that's a fancy $3 word for we make a copy of it. Okay? Now, if you think for a second go back to our library analogy, if you had a very rare book that was very important, and a high school student needed a piece of this book for his research project, would you give him the book? No, you'd make a photocopy. And your cell does the same thing. You make photocopies, if you will, of the genetic information so that it can be used in the cell. And then the part of the number of photocopies that are made, the rate at which they're made, the rate at which they're destroyed, all of that is important in determining all of the things about the way that the genes are expressed and the way that the proteins are made. So if I'm making a copy, if I'm in a library and I'm making photocopies of something useless, I'm going to get fired for wasting ink and wasting paper. And that's also true in a cell. If I'm making copies of stuff that's useless, I'm wasting energy, I'm wasting raw materials, I'm not as likely to reproduce. I'm likely to grow slower and be outcompeted by other organisms that don't make copies of junk. Okay, so this is a good evidence that this is not junk. Another reason is because a surprising amount of DNA is conserved, and that's a $3 word that means it's the same in a lot of different organisms. And even an evolutionary biologist will tell you that conservation indicates function. So this is not me arguing this as a creationist. This is standard biological theory. If I'm transcribing it, it probably is functioning. If it's conserved, it's probably functioning, right? It gets even more complicated because the DNA that's transcribed, at least a quarter of the entire human genome is transcribed in both directions. So DNA is anti-parallel. It has one message one way and one message the other way. And you'll have to forgive the slight meaninglessness of this sentence because it was the best I could do to make a sentence that said something different backwards and forwards. But this sentence, if you read it forwards, says strap on no pets. And if you read it backwards, it says step on no parts. Okay, so you have two meanings out of the same sequence of letters. Now think about if I make a change here, right? I'm going to make a slight change. Strap on no pots, stop on no parts. I've changed both meanings. Okay, so this is going to be important for our next question about whether mutations can be neutral. So if I'm reading the DNA in both directions, if I have one change, I've changed two different sequences. That means I've changed two different regulatory elements, I've changed two different proteins, I've changed two different something else. And in many organisms, you can transcribe a particular region of DNA and you can cut it into different pieces and you can use those different pieces in different ways. So the same sequence can code for 3 or 4 or 5 different things. So when you talk about changing something in the genome, you have to think about the fact that this is very complex, it's very interdependent, and a small change actually has a very large effect. So it turns out that basically all the things that we previously identified as junk are useful. So evolutionists might object and say, well, pseudogenes are useful. They're things that look like genes but aren't. Well, it turns out that pseudogenes are very important in regulating gene expression. All of the stuff I was just telling you about, when you turn genes on and off and how much you have. Well, the stuff in between the genes, that's not important. No, that's where you have promoters that start transcription and repressors that stop transcription and nucleosome binding sites. So you've got these little proteins that the DNA wraps around, because if you spilled out all the DNA of a single cell, it would stretch from the floor to slightly above my head, because I'm not quite 6 feet tall. But all that is contained in a cell that's so small that you can't see it without a microscope. So you have a very long piece of spaghetti, and you have to be making copies of parts of it sometimes, and parts of it never, depending on where it's located in your body. So in every single cell, part of your DNA is wrapped up really tight on those histones, those nucleosomes, and part of it's wrapped up loosely so it can be used. And if all of the regions in between the genes determine how that wrapping happens. So if I change a region and it changes the way the DNA is wrapped up, that means something's going to get locked up that needs to be accessible, and something's going to be open that needs to be locked up. So those are very important things. Transposable elements, these are sometimes called selfish genes or jumping genes. They sort of move around in the genome. They actually have multiple functions, one of which is chromosome pairing and DNA repair. And DNA repair is super important because, as we're going to get to when we discuss whether mutations are good, if you have changes in your DNA, it can be very bad for you. And we know this based on if you just look at environmental policies, right? We're trying to eliminate as many mutagens from our environment as possible. Why? Because mutations are bad. All right, repetitive DNA is another thing that evolutionists will say is useless. Well, it turns out that your chromosomes, your genome is split into 23 different pieces, roughly 46 different pieces, really, if you count both sets of chromosomes. And each chromosome is capped on the ends by repetitive DNA called a telomere. And we think now this is what keeps you from dying and determines how quickly that you age. Because when your DNA is replicated, when a copy of it is made, it doesn't go all the way quite to the end. So you lose a little bit of your telomere each time you make a new cell. And eventually you get to a point where you can't lose any more, and then you die. And that's the current theory as to why people age and die, and they can't live beyond about 120 years, because your telomere just eventually gets so short you can't make new copies of your cells. So I wouldn't call that useless, even though it's repetitive. So I think all of the genome is useful. Even if only 80% of it is useful, it makes it very difficult for evolutionists to explain how we have the mutation rate that we have, and we don't have genetic entropy, we don't have error catastrophe in the genome. So turns out, not that much DNA is useless. So we've invalidated the first proposition that we need. And I could stop here and say I've made a pretty good case against mutations being a good mechanism for evolution. But, you know, even though this is kind of like swatting a fly with a Buick, I'm going to keep going, and I'm going to demolish all 4 points. So the second question is, can I have mutations that are neutral even if most of my information is useful? Okay? So I'm going to use an analogy again and give you a short piece of DNA that has a very clear message. Now I'm going to mutate it. And if I sent you this, you might think, well, she doesn't proofread her text messages very well, but you would still understand that what I'm trying to tell you. And you might think I'm from the University of Steubenville, because they say that there are a lot. But if I add one more mutation, now you think that I'm trying to explain to you in very broken English that I understood what you just said. Very well. Okay, so if I take those 2 mutations separately, got it good and got is good, you probably would get the original meaning, that I was trying to say God is good, and I made a mistake. But when I put them together, they're no longer neutral. They've now altered the meaning of the sentence. So, can a mutation truly be neutral? Well, it can fly under the radar for a little bit, but once I start adding additional neutral mutations, I start really damaging information in the genome. So an objection that evolutionists might raise to this is the idea of silent mutations. So when you have a genetic code, it's coded in sets of 3. These are called codons, because scientists are not creative and they name things exactly what they are. So the last base, the last letter in the codon, is sometimes called a wobble position, because certain codons can code for the same amino acid. So technically, in this case, I would have a short piece of DNA that codes for the same thing that goes into the protein. So even though the letter has changed in the instruction manual, nothing has changed in the protein. So you think, okay, well, at least this can be harmless, right? This can be totally neutral because I'm not making any change in the protein. Au contraire. In a recent article published in Nature last year, and Nature is one of the most well-known and well-read biology journals of all time, right up there with Science. My PI when I was in graduate school desperately wanted to get published in one of the two, and he just couldn't quite make it. But in an article that's published last year, researchers looked at 8,341 yeast mutants with mutations in 21 different genes. We would call this a significant N value. They've used a large number of mutations in a reasonable number of different genes. And they determined that 76% of silent mutations were significantly deleterious. They slowed down the growth and replication of the organism by 1% or more. And that doesn't sound like a lot, but when we get to the very end, I'll show you what 1% decline looks like over 100 generations. You get really, really low really fast. Why? Because every nucleotide carries a little bit of information in a way that we can't even wrap our minds around. So even if it's coding for the same amino acid, it might be a rare tRNA that isn't as present in that organism, because organisms have codon bias, so they prefer one over the other, even though technically they code for the same thing, right? It might slow down replication. It might cause the DNA to stick a little bit harder at that particular spot, because you have to open up the DNA, the two strands of DNA, in order to make a copy of it. So if it's sticking a little bit harder, that slows down your replication. If it's a rarer codon, that slows down your replication. If you slow down replication or transcription, if you slow down the making of a copy of the DNA, you slow down all the cellular processes that are associated with that copy. And even if it's just a little bit, that actually decreases the fitness of the organism overall. So even mutations that are supposed to make no difference at all, actually, when we look at them on the record, 75% of the time are bad for you. There are no neutral nucleotide sites. So by the very fact that it exists, a nucleotide, it affects spacing between other nucleotides. So that is going to affect the speed of making copies. It affects like I said, that regional nucleotide composition, how sticky those two pieces of DNA with each other. It affects how the DNA folds. Remember, I just talked about the histones and how it's wrapping up, and how sometimes it gets wrapped up really tightly and sometimes it should be wrapped really loosely. It affects how the copy of the DNA folds, how the RNA folds. And the three-dimensional structure of the RNA is going to affect how the RNA that information is translated into a protein. It's going to affect whether the RNA just balls up on itself and can't be used. It's going to affect all kinds of different things. So Dr. Sanford said it very well when he said if a nucleotide carries even if it carries absolutely no information, it's not involved in any of these processes, it is by definition slightly deleterious, because having it there without it doing anything at all would slow cell replication and waste energy. And over time, the individual that has that nucleotide is going to die faster than the individual that does not. And the population is going to favor the individual that does not have the useless nucleotide. So none of the genome is junk. None of the changes can be neutral. They have to be good or bad. Okay, so the next question is, can they be good? According to Dr. Richard E. Lenski, who's pretty famous for starting an experiment in 1988, which is still ongoing, involving evolution of E. coli, which I could give a whole presentation about by itself. I'll just address a little bit here. He says perhaps 1 in a million mutations are good. So he's already helping me with my argument if you take what I've just established, which is that you can either be good or bad. You can't really be neutral. So we've got one good mutation to 999,999 bad ones. I don't think we're going up. okay? But let's talk about, can you even have a good mutation? Because I'm going to make the contention that I don't think you can, okay? But first, a little bit about Linsky. And Stanford deals with this very well in this quote from Genetic Entropy. And he says there's a few isolated cases, and he's referencing this one in particular, where it's claimed that certain laboratory experiments demonstrate remarkably high rates of beneficial mutation. The key to understanding these claims of superabundant, high-impact, good mutations is that a large part, up to 50% of any microbial genome, consists of just-in-case genes which need to be precisely regulated. These genes are crucial in the real world when you have real environmental conditions that are changing constantly. In the laboratory, you have a constant food source, you have constant temperature, you have constant aeration, you have constant humidity, all of these things are kind of kept constant that would change constantly, even over the course of a day in the real world. You know, as think about your backyard. Some areas are in sun for part and in shade for other parts. The conditions affecting a microbe are going to vary drastically even throughout the day. They're not varying at all in the laboratory. Okay, so in an artificial and changing laboratory environment, these genes that the bacteria needs just in case it gets too dry or it gets too bright or it gets too hot, they're dead weight. And remember what I said about those nucleotides that are if they're being copied, if it's sitting there doing nothing, it's bad for the bacteria. So if I have bacteria in a laboratory and I break one of those genes, or I break the regulation of that gene so it's no longer getting copied, that bacteria is now evolved in the sense that it is more fit in the laboratory environment. But if I put it back out in the wild, it's missing something it needs. So this is a good way to interpret this kind of experiment. But in case you know anyone would be worried about bias towards creationism, these guys are not creationists, and they basically said the same thing, that these data represent the distribution of fitness effects of extremely maladapted populations. These are populations that are not well suited to the real world, that are important only in the artificial environment of the lab. So it's pretty well consensus that these laboratory experiments show you what happens in a laboratory, not what happens in real life. So Linsky aside, mutations are not good. If you know anyone who has a mutation, it's probably a health problem for them. There are approximately 10,000 different human health conditions that are related to changes in a single gene. I certainly don't know 10,000 beneficial conditions in humans that are related to changes in a single gene. And then, of course, every research paper that you read that discusses mutations, it immediately starts out with the vast majority of them are not beneficial. You know, the vast majority of them are not good for you. And in case you're wondering what this graphic is, this is lineages of COVID-19, starting in December of 2019 and up through April of 2020. And everywhere that a line stops, that means that strain stopped replicating. It stopped being found in the environment because mutations are bad for you. As the coronavirus was mutating, yes, we had a few new strains that circulated a little bit better than others, but mostly we had strains that just went extinct. So science tells us over and over and over and over again that mutations aren't beneficial. But occasionally, evolutionists frequently will pick particular instances and say, well, this one's actually a benefit. And antibiotic resistance is one of the major ones. So I'm sure you've all heard of MRSA. That's methicillin-resistant Staphylococcus aureus. If you go to the hospital, MRSA is a serious concern. And it's argued that, MRSA is an example of a beneficial mutation. So this is great for the bacteria, right? In the presence of the antibiotic, it won't die. This is awesome. Well, methicillin works by binding the cell wall of Staphylococcus aureus and causing that cell wall to break apart, disrupt. So MRSA has, instead of having sort of straight, strong connections in the cell wall, it's slightly wonky. So if you think about molecular things, shape is always important. So methicillin can bind this, it can't bind that. So that's why the MRSA don't die. But this isn't very strong compared to this. So if you put MRSA in a test tube with non-resistant Staphylococcus aureus, the MRSA dies. It gets outcompeted rapidly by the non-resistant Staphylococcus aureus. So if you remove the antibiotic, having that mutation is not an advantage because it has actually weakened the cell wall. Now there's other mutations in similar bacteria that work by, they weaken the ribosome, or they weaken, they cause bacteria to chuck out things that they shouldn't be removing, but it happens to also check out the antibiotic, so antibiotic resistance is really no help in general to evolutionary biology. It's no help in particular for MRSA. So I call this the spork problem, right? You can't actually make something better about a spoon or a fork without making something slightly worse. So I wouldn't want to eat soup or spaghetti with a spork. But there are a lot of things that I could eat with a spork, but I'm not going to be able to eat any of them as easily as it would be if I had the proper instrument. And this is true even at a molecular level. So if I get a change that sort of seems to benefit in one situation, it's probably going to make me less well adapted to another situation. And I'll tell you, I've been doing this for 8 years. I've been looking at individual claims of beneficial mutations for 8 years. I have never found one that doesn't have a spork problem. And I could literally spend 3 hours talking about all of the different ones, but I'll give you a couple more, just in case. Tusklessness in elephants was claimed to be beneficial because if an elephant doesn't have tusks, it won't be poached. So it'll survive longer, right? Well, turns out the tuskless mutation is paired with an X-linked recessive lethal gene. This means 2 things. Only females are tuskless. That means that the males have a higher likelihood of getting poached. And if you know anything about genetics and gene pools, if I kill all of the males or most of the males in a population, I'm rapidly going to have a very inbred population and I'm going to have extinction. Okay, half the male baby elephants that are conceived die in utero because they get the tuskless gene with the X-linked lethal gene and they don't have another X chromosome to cover that gene up and allow them to survive. So not only am I going to poach more of the males, I'm going to have half as many males, and the fertility of these female elephants is about 67% of what the fertility of regular female elephants would be. This is not a beneficial mutation. This is going to lead to extinction. It's not going to lead to elephants being preserved. Sickle cell trait is often mentioned as a beneficial mutation because having one copy of the sickle cell allele is protective against malaria. Now, it's a survival rate of 86% versus 84%, which is kind of impressive, but it's not that impressive when you take into account the fact that if you have 2 copies of the sickle cell allele, you have a 90% chance of never reaching adulthood. So if you have 2 parents that each have a copy of the sickle cell allele that gives them a 2% higher chance of surviving malaria, and they have a baby, they have a 1 in 4 chance every time they have a baby of that baby not making it to adulthood. In addition, even having the 1 copy is bad for you if you're doing heavy exercise, if you're dehydrated, or if you're at a high altitude, because your blood just doesn't carry oxygen as well. And honestly, the sickle cell trait one kind of drives me crazy because you have a bunch of American intellectuals or European intellectuals claiming this is a beneficial trait when there's a bunch of kids in Africa that are dying from it. Right? This is actually.
Well, that's why they think it's beneficial.
Perhaps, but it's ridiculously racist, I think, to think that this is beneficial, because the only way that you could think that is if you weren't actually being affected by the devastation that this mutation in the population causes. Okay, so while we're talking about mutations and benefits of mutations, I need to throw in a little bit about the idea that mutations can generate new information. So you'll often hear evolutionists make claims like, oh, there was a change in the genetic sequence, that's new information. Technically, that's not incorrect. However, that is not the kind of new information that you need to go from a microbe to a man. This is the kind of new information that you need to go from a microbe to a man. And no evolutionist has yet shown me an example of a mutation that generates that much new information. So you've got God is good at the top, and then the 6th question of the Summa at the bottom. And you'd actually need twice that much information to go from a microbe to a man. That's how my analogy limps, because there's not enough space on my slide. So when we're talking about mutations being a benefit or mutations giving new information, you kind of have to keep this image in mind. Okay, am I making this much new information? Well, evolutionists claim that they've had some examples of new information, just like they claim they've had some examples of beneficial mutations. And the best one ever is from Dr. Barry Hall, and he documented evolution of the EBG gene. So let's look at this for a moment. This is going to be the most technical part of the presentation. I'm getting towards the end, so I have to talk really fast, so I'm sorry. But he knocked out, so he basically was able to break the gene in question. The ability of the E. coli to produce the enzyme lactase. And lactase is an important enzyme that allows you to digest the sugar lactose, which is present in milk. So if you don't have lactase, you can't digest lactose. So basically he made E. coli that could not live in lactose-only media. And then he put them in lactose-only media to see if they would evolve. And they did. Mutants arose much more rapidly than he expected, which actually is a warning flag. If something is arising much more rapidly than expected, it probably means that the machinery for it to arise was already in place, and we're not looking at actual major evolutionary changes. So he found out that the ability to metabolize the lactose sugar, to live on the lactose, came from a mutated protein that he called EBGA. And then he looked at the original unmutated protein, which he called EBGO. So he said, okay, this is the protein that causes them to be able to to live on lactose. I'm going to look at the protein before it got mutated to see how it evolved, right? The unmutated enzyme was able to break down lactose in a test tube. So it already possessed the ability to break down the sugar that it's now breaking down to allow the E. coli to survive. And remember what I said about gene regulation, things getting turned on and off and certain times? Okay, what's happening here is not a new gene being created or a new function being created. This enzyme already had the function that is now, giving the E. coli the ability to survive. So it had a pre-existing ability to metabolize lactose. But this is the best new information that Dr. Miller could put in his 2008 book, Finding Darwin's God, and this experiment was done in the 1970s. Okay, this is the best they've got. And even Dr. Hall said, and I'm going to read this in English instead of in the lab language it's written in. I'll start with the second sentence. All of the new functions evolved for the enzyme that we looked at represent variations upon a general function that it already had. The unmutated enzyme doesn't break down any of these sugars that we tested at rates fast enough to keep an E. coli alive, but it can break down all those sugars when we test it in the test tube. So it possesses a pre-existing ability to break down those sugars. In a real sense, the mutations that generate the evolved enzymes do not result in truly new activities. They simply improve old functions. And I could, again, do a whole other presentation of exactly how that works and why that works and why even that is not even remotely close to evolution that we would need to get from a microbe to a man. But I was told to end 5 minutes before Mass. I have 4 minutes left. I'm not done yet. Sorry. But you can clap all you want. No, I'm kidding. The last thing I want to address, that fourth point, can we get changes? Can we get enough good changes that so we've already kind of like knocked this out, right? Like there's no neutral changes, there's only bad changes, there's no good changes. Okay, but could we get changes accumulate to create new kinds of organisms? Again, a small biology lesson here, right? A species is an individual type of organism. So this is a red fox is a species. A dog is a different species, even though the fox and the dog are similar. What do we mean when we say create new kinds, right? Kinds, depending on the taxonomic classification of the organism, fall somewhere at the genus or the family level. So all of the dogs will fall under the same family. So wolves and foxes and domestic dogs and anything else that looks dog-like. But we've never observed ever in any laboratory experiment that's ever been done a new, or in anything happening in the wild, a new genus forming. Okay, so we've never observed a new genus forming. That means we've never seen something like a snow leopard give rise to something like an ocelot. Okay, so if you understand kinds intuitively, as you know a cat can't give birth to a dog. Well, this kind of cat can't even give birth to that kind of cat. We can't even observe that. So do changes accumulate to create new kinds? Well, a lot of people will point to speciation and say, well, this new species has arisen or that new species has arisen. And one of the things that's brought up a lot is the peppered moths and the changes that were observed in the peppered moths as London became slightly more smog covered. There was an experiment that attempted to show that the population shifted from light colored to dark colored moths, and the guy who did this experiment would have failed my research methods class for all of the things that he did wrong in his experiment, like taking the moths out in the morning and warming them up over the hood of his car to get them to move enough to like get onto the trees, and then he just put them on the trees and the birds just picked them off like it was a feeding tray. There's so many problems with this experiment, but even if he was right, even if he showed that a light colored moth turned into a dark colored moth, the title of the slide shouldn't be, is speciation a good example of evolution? It should be, is this even an example of speciation? Because a new species means that it can't interbreed with the original species. These moths can interbreed, and there's still light-colored and dark-colored moths all over England. Right? Darwin's finches, which are supposed to be 14 different species, can interbreed to the point that there's a maximum of 6 different species, maybe only 1 different species, because they haven't done all the experiments they would need to determine that yet. Mallard ducks can breed with other species of ducks, and even geese. These are not different species. So when somebody says, oh, but we've seen all kinds of new species, ask them to show you one. One solid new species that has been developed. I bet they can't do it. And even if they can, they can't show you a new genus. They can't show you a new family. They can't show you a new class. They can't show you a new order. They can't show you a new phyla. They can't show you a new domain. So how can you possibly say that I went from a microbe to a microbiologist if I can't even show you how I go from a moth to a new species of moth? Okay, so over time, all the experiments we've ever done, no matter how much we mutate them, bacteria stay bacteria, fruit flies stay fruit flies, peppered moths stay peppered moths, and finches stay finches. And even the species stays the same. The Drosophila that's mutated in the lab is still Drosophila. It's not even a different species of fly. So even evolutionary biologists will admit this when pressed. So this is a proponent of evolution here. Pierre de Grasse had this to say: in some, the mutations of bacteria and viruses are merely hereditary fluctuations around a median position. So there's like a norm. And they'll swing a little bit to the right, a little to the left. There's no final evolutionary effect. Why? Because there's no room in the genome for the kinds of changes that evolutionists routinely posit as a source of variation for all living things. There's no junk DNA. There's no neutral mutations. There's no beneficial mutations. And there's no new kinds. Case closed.